And so they just more or less put a guarantee on it. That's why you need your vitamins. Okay. Let's first start by understanding the purpose of cell respiration. Hopefully we do understand that cell respiration is going to be having a purpose of converting chemical energy. From one form to another. You have energy in glucose. You react it with oxygen, so you're combusting it. And much like we combusted the potato chip, your products are going to be CO2 and H2O. However, there's a ton of energy in glucose, and there's low energy in your products. Yes, and then there was also the iPad too.
That was iPad.
And then the slides that are on the actual slides. But don't worry, we'll take care of this tomorrow morning. It's just best we do in the morning.
Thank you.
Mm-hmm. Low energy and then high energy for the glucose. Okay. So the question would be, where is the energy? Where's the energy? Where's it gone to? Right. So we've also made some ATP over here. So maybe the best way to view this, when you talk about energy on the planet, you've got plants, and the plants are basically doing photosynthesis. To make sugars and other organic molecules. Those sugar and organics, those organic molecules are taken in by animals, which do respiration. The two products of respiration, H2O and CO2, are what feed the reactions in plants, the photosynthesis. And along the way, bless you, the animals are producing the high energy molecule ATP, which of course is what powers any process in a body. Now, the other thing to remember is that every step along the way, it's unavoidable, it seems, that we produce a ton of heat. So a ton of heat is produced along the way, which is what makes the system overall having an overall inefficiency. I'm sorry? Yes, basically. Because all these processes, any energy conversion process is going to result in a loss of heat. Yes, ma'am.
Hi, this is a slight tangent, but it deals with this. So when plants, they use photosynthesis, and instead of like saying animals that take in respiration, could you just say plants take in respiration as well?
They do.
Okay, so it's like you could pretty much have that same little chart for just plants.
I could, yes, absolutely. And in fact, your book has one that's a little bit stronger. And I encourage you to like draw out, or definitely be reading the book. But as you look at the book, you'll notice that this picture is drawn in there, but instead of saying animals, plants, one or the other, because as you just pointed out, I'm deficient in that I've said that animals are doing the respiration only, and the reality is that plants are doing respiration as well. your book actually just calls it chloroplast, mitochondria, and they consider them the two reactions of each. And they don't stick a reaction with a certain type of creature. They stick a reaction with just a certain type of organelle. And that is an important point to make. That is such a, I couldn't begin to tell you, it's such a problem for people coming out of biology in the ninth grade, is they think that chloroplasts are for plants and mitochondria for animals, and that is it. And if you believe that, you are extraordinarily wrong. And that's the only way I could ever put it. It is extraordinarily wrong. Plants also have mitochondria as well, and we really have to get that. So that's a good point to make, and I'm glad you made it now, rather than me waiting till the photosynthesis chapter to kind of drop that bombshell. Let's talk a little bit about the role that catabolic reactions are playing in this. What is a catabolic reaction or catabolic process? Okay, so it's a reaction that results in a breakdown. So let's break it down. Catabolic reactions. And one thing we see with this breakdown is that there's always a release of energy. And typically that energy gets put into two spots. One place it gets put in is it gets put into ATP, we saw. The other place it gets put into is heat. So the energy ends up in the ATP and the heat. Now, when we look at respiration, there's really two different ways we could go about doing this. We could do one route, which is going to be the poor route, fermentation, where fermentation results in essentially a very little amount of ATP produced. And also, it's a non-combustion based reaction. It is what we would call anaerobic. And this opposes the aerobic respiration, which produces tons of ATP. By combusting
And would fermentation be using like bacteria
with
versus
oxygen.
animals?
Kind of. You are completely right on that, except that we sometimes do fermentation as well.
Okay.
And to put a time and place on this, because I think it's really pivotal, fermentation is going to be outside the mitochondria. So certainly not within. We can at least say fermentation is not in mitochondria, whereas aerobic respiration is in the mitochondria. So maybe we're getting that mitochondria means that I could be doing some aerobic stuff and producing more, and lack of mitochondria means I would produce as little. Now, right away, this puts an important cap on the size of creatures. We know that typically creatures that are doing fermentation only are very small. A fungus and a bacteria would be two good examples of doing the fermentation only. And so we also want to be cognizant, we want to be set with this idea that fermentation would result in a very little amount of ATP being produced, and that very little amount of ATP is being produced outside the mitochondria, certainly not within. Okay. your book actually would, instead of writing ATP, it would write just energy being produced, and then you have to understand that energy is stored in ATP. We might as well write energy as well, I think. Since you're taking C6H12O6, a pretty complex molecule, seems like you're just shattering it into a bunch of little pieces of CO2 and some H2Os along the way. So you can tell you're taking like this massive wall basically and smashing it down, and that energy that was in the wall ends up in all these little components. Whatever energy is lost, is hopefully lost, is energy that can form ATP, not heat energy. Now, one problem that people have initially understanding this is they recognize C6H12O6 as glucose, or maybe fructose, maybe galactose. But the question they might have is, well, what if I'm on like an Atkins diet? An Atkins diet is a diet where you don't ingest hardly any sugars. You ingest almost all protein. See, the deal is that people that aren't ingesting glucose, if you have no glucose, you still are able to do a process that's similar to this. And the reasoning behind is because if you take in any type of sugar or any type of protein or even fat, there are reactions that can take each of those and can feed it in by converting those chemicals to glucose or feeding them into the overall process of respiration. So what I'm suggesting is that somehow these other food types that you're eating, the fats, the proteins, and the other sugars, the other carbohydrates, they're either converted to glucose or they're converted to some derivative in this process, which puts a guarantee on the fact that I could break down fats, proteins, or sugars. Now, the problem is that these are all enzyme-based reactions, and so some enzymes are more prevalent and some enzymes are more strong, or they are stronger. So if you think about a lot of the enzymes for proteins and sugars, they work very effectively. However, some enzymes for a fat breakdown, not so effectively, which would explain why it's unlikely to break down fat, but it's more likely to produce fat, because the difference is, or the reality of it is that every time I've drawn this purple arrow here, I could actually draw it to the reversal side. What does it mean if I'm reversing this arrow like I'm doing right now?
That you can convert to C6H12O6 like the fat.
Right, you can interconvert between all of these. And what you end up with is like this massive like series of interconversions that could exist and do exist when we're looking at all of metabolism. I'm going to show you guys a little reaction, or a little map real quick. It's actually kind of it's a cute little thing. If you ever were to major like in biochemistry, this would kind of be what you would be doing all the time. At least an understanding of this map. Tell you what. I think I want to save this. I want to put it out. For all the doctor wannabes out there, this you have to understand before. You have to understand early on, because you'll be taking a class called biochemistry, and in biochemistry you would actually spend a fair amount of time discussing this topic. This always puts a smile on everyone's face. Yeah. Yeah, it is, isn't it? I mean, I think we're going to get it. Our focus today, actually, is going to be right through about here. Let's see where we at. Definitely, one thing we can understand is where is and this box is very, very pivotal. We'll talk about it in just a second, but this rectangular box I've drawn, describe its position on the paper. Kind of in the center. And maybe one thing we're gathering a little bit is that all these little side reactions you're seeing over to the left top, right bottom, every which way, it seems like, and I'll tell you the rest of the story is that tomorrow we'll actually continue talking about some of the reactions that are down this direction as well. So one thing you can get is that we're kind of feeding any of the members to your left or to your right into this central process. And the central process is respiration. So all of metabolism would be everything on the page, but respiration would be everything that is only in this area. So these are non-respiration based reactions, but they're conversion reactions that convert between other things. Like for instance, and this is a little hard to see, but this is like very out of focus. Here it looks like you have some reactions of different vitamins, interconversion of vitamins. Where are your amino acids at? Some of your amino acids are down here. Looks like some are right through here. You can convert from one amino acid to the other. Y'all remember what amino acids are, right? What do they make up? Proteins. So you can ingest just about any food type and you can convert it to an intermediate, at least, in this global process. Now today, my focus is on that initial part of the process. That initial part is going to be the part that would be anaerobic, the part that would be occurring without the need or without the input of oxygen at all. But before we do that, one thing we have to establish is we have to understand this idea of reduction and oxidation real quick. So let's talk a little bit about what reduction and oxidation are. Let's talk about redox reactions. The example from the book I had never really paid attention to till I was asked this morning about it. That's a horribly bad example. So I wouldn't really stress the example from the book too much. When we look at reduction though and oxidation, that's kind of where the redox term comes from, reduction. Reduction always results in the gain of electrons. Oxidation occurs when there is a loss of electrons. But sometimes in biology it's quite difficult to follow the movement of electrons. So the way I would remember it, if you're starting out in biology and you haven't taken biochemistry just yet, is to remember that for biology, reduction is when you have the gain of hydrogen, and oxidation is when you have the loss of hydrogen. You remember those two things, you'll be pretty much set. A way that I remember is by giving just a quick example. If we take Na and Cl, both of which have a zero charge, and they react. Since Na is a metal, Na gives its electrons to the Cl, so Na becomes +1. The Cl becomes -1, based on the give and take of electrons. See, the deal is that the sodium has 1, the chlorine naturally has 7. So the Na's 1 electron ends up on the Cl. The Cl still has its own individual 7. That's why it's 1 minus 1 plus. Cool? So in chemistry, we would have learned that the Na was being reduced or oxidized? Oxidized. And the chlorine was being reduced. Now there's the example from like chemistry. But another example that I'd like to focus on would be NAD plus and NADH. So here's what the deal is, and this is more biological than it is chemical now. I'll describe the reaction, then we'll take it apart and see exactly why we're doing this. NAD plus has the potential to gain 2 protons. Actually, it's just going to be 1 proton in the end. And 2 electrons to become NADH. That would make sense. NAD, there's the H, 1 H, 1 NAD. That would be 2 positive plus 2 electrons, results in a 0 overall charge. That would mean that this right here is the reduced form, which means that the process of reduction had occurred. So what if I undid it to show the oxidative process? If I took NADH and I formed NAD plus out of it, and it lost its proton, its hydrogen, and its 2 electrons, this would make this process oxidation. Now, everyone was pretty much cool when I did the NaCl part, and now I'm over here doing NAD, and so a lot of people say, well, why this NAD thing? Because it's certainly not like periodic table based like the Na and the Cl were. So let's talk a little bit about why we would talk about NAD or NAD+. First off, what does NAD stand for? That's a good question. It's a nicotinamide. It can exist in its oxidized form or in its reduced form. And for our purpose, it is our electron carrier. I had drawn out a little something the other day, and that little something went like this. And in this example, I had alluded to how glucose was this high energy molecule and how carbon dioxide was the low energy molecule. And along the way, you were doing a series of steps that resulted in the removal of protons. So there's all these H pluses coming off, as well as a bunch of electrons. And as you pull off these electrons, as well as protons, you have to store them somewhere. And we collectively store them by taking NAD plus and reducing it to NADH. So basically you could view the NADH as basically a storage molecule. A better analogy though for real life would be that the NAD is like a bank account. Every time you make a paycheck, you don't go below it all. You save some, and then you save it for a moment where you basically spend it all, whether it be a new car, a new whatever it is you guys are buying these days, the new Justin Bieber CD from me, or whatever your life like holds for you. You store it in your bank account. You cash it in eventually. Cool? Awesome sauce. So, I guess hopefully we're getting that somehow we've got to be taking glucose and breaking it down in a bunch of, do you see me does glucose just jump off? going step by step. So we need to go over the steps. You know what I've drawn there? Mitochondria.
Looks like calligraphy. Why did you roll the lines?
What is calligraphy? Mitochondria. It has two membranes, right? The outer, and then what is this one called? The inner. What's the name of this space in the very, very middle? The Oh, I'm sorry, in the very, very center, my bad. The matrix, and then this is called the inner membrane space. And so what would this area outside be? The cytosol. Cytoplasm is the area. Cytosol is the fluid. So I could have said either which one. And let's start off with like our basic ingredient. And for this process, what was our basic ingredient? So we are starting off with glucose. Now, through a series of 10 processes, one little arrow, two little arrow, three, dot, dot, dot. There's more arrows along the way. The glucose is converted to 2 molecules of pyruvate. Pyruvate. Now the 2 pyruvates that are produced are each 3 carbons long. So I'm going to use the orange here to represent like the number of carbons in each molecule. We know glucose is 6 carbons. The pyruvates, though, are each 3 carbons. Have I conserved carbons? 6 at the start, 6 at the end. 100% conservation. But you didn't conserve all of your hydrogens or all of your electrons. Not only did you produce 2 pyruvates, but you also produce 2 NADHs. What did NADH represent earlier? It was like your bank account, right? It's not ATP, but it's potential. The other thing I like to use for an analogy as well is like when you have NADH, that's like having a statement from your bank that says that you have some money in there. If I go up to Best Buy, and let's say my checking account has $3,000 in it. If I walk up to Best Buy and I show them my Chase bank account and I say, I want this new TV. It's $500. It's on sale. Here's my bank account from Chase. So could I just take this now? Is that the way purchases work? What do you do instead? You give them the money, right? You burn the ATP. The NADH just represents that you could make some ATP. Just like a bank account statement means that you could make some money. You could if you go to an ATM, I guess. It's potential, though. So some potential energy. The pyruvate could still be broken down, though. And also, to start this process, you had to spend a little energy. So you have to invest 2 ATP, which right away seems like a shoddy thing because if you're investing ATP, this process is supposed to produce me some ATP. That's like going up to the Chase bank store or whatever it is and having to like pay them money to open a bank account. Typically you give the bank money so you can have an account. You don't have to pay money to open an account. So you're having to invest in a little bit of ATP initially, but fortunately through this overall 10-step process, you produce 4 ATP. So what's my net ATP production? So this I remember is the rule of 2. 2 pyruvates, 2 NADHs, 2 ATPs. Works out perfectly.
This is
Correct. This is an anaerobic process. Because it's in the mitochondria where you can be aerobic. Which means oxygen's not used at all. So you had 10 steps that took you from glucose to pyruvate. The name of those steps is glycolysis. What does the glyco prefix stand for? Sugar? What does lysis mean?
Breaking down.
Or split? This is the sugar splitting reactions. What does it mean to split something? Cut it in half, maybe? You took glucose and you basically cut it in half. Cool? Now your pyruvate has one of two options. These two pyruvates could do one of two things. They could be drawn inside into the depths of your mitochondria, or you could do a couple of different reactions to keep you on the outside of the mitochondria. Which do you think would be best? Because you're able to be aerobic. Aerobic means I have oxygen. Anaerobic means I have no oxygen. If you're breathing, we're going this way. If you're not breathing, or you have a cellular lack of oxygen, you're going this direction. This direction would be the fermentation direction. And in fermentation, you produce one of two products depending on what type of creature you are. One of the products you could produce is lactic acid.
Isn't lactic acid the stuff that makes your arms red?
Yes, it is. Now the cool thing at least is that you take your two NADHs that you had produced before and you remake them into two NAD pluses. Do you know where those two NAD pluses can go? They can go to the first part of this glycolysis reaction so that you can end up remaking them into two NADHs. See, you're recycling your NAD plus there so that you could reuse it because you've already messed it up. One thing to mention that we'll get to soon is that NADH gets cashed in way down the line over here. If you're not going into the mitochondria, then you have no need for the NADH produced at this point. So we might as well reverse the process. Yes, ma'am.
What is the purpose of fermentation? Like, why does it produce lactic acid?
The lactic acid or ethanol that is produced is basically the byproduct of the anaerobic synthesis of pyruvate. Essentially, this lactic acid that is produced, and lactic acid first off is produced like in animals, whereas the ethanol is more of a bacterial thing. But the reason you produce the lactic acid is because it's the product basically. It's what happens when you oxidize the NADH to form NAD+, and by the way, that's an oxidation reaction right there, whereas this had been a reduction reaction to produce the NADH. But it's all about remaking the NAD+ so that you could do more glycolysis. Because if you think about it, if we just said, you know what, the pyruvate can't go inside because there's no oxygen, so I'm just going to let the pyruvate sit here, and let's just say fermentation had never evolved as an option, then you would have no NAD+ to redo this process. You'd be stuck. If this is somehow an issue, like in understanding, please, one, do one of two things: watch the video again, because it just somehow seeing it again helps, or read. Dude, this is all in your book. I promise you. Promise you. Okay. So the lactic acid. Let's talk a little bit about the lactic acid. It's what hurts your muscles, right? We can agree that this hurting of the muscles is something that you wouldn't want to feel. If you've ever ran before and ran too much, you know you produce the lactic acid. Describe to me what happens when you do that. You've ran, it hurts. Tell me, where do you grab? Sometimes you grab your muscles, but I want you to really think hard about your life. Where does it hurt the most if you've been running way too much? Hurts right about here. What's here in the body?
Where?
Right here, and sometimes in the back. Your kidneys, your liver. Hurts there. And that's because your kidneys and liver are able to take the lactic acid and reconvert it into glucose. Now, as they do that, think about it. Breaking down glucose produced energy. So what do you think taking lactic acid and reforming it into glucose would require? So it's going to require some input of ATP. So the actual feeling that you're having is you put your hand on your side and it hurts, and you realize that cramp that you feel there isn't permanent. If I ran when I was 5 years old and this happened to me, I'm still not holding right here. That feeling you're having is for two reasons. One is that you have an acid going to your liver, and two, your liver is like squeezing itself, trying to empty itself of any free ATP, any free glucose. You're having to expend or expend energy so that you can reburn your glucose and hope that this time you could be more aerobic. When you talk about this, I haven't worked out in months or years, it's time for me to start doing it. Do you think that people that start that process up, do you think they feel good the first time? The reason is because they don't have any endurance, right? Isn't that what we call it, endurance? Well, the reality of it is that the endurance that we're discussing, this endurance really isn't, it's not really endurance at all. Endurance is this idea in our head. It's like it's mental. It's really not. It's biochemical than it is anything else. If you have endurance on a cellular level, you can go right on through this process. Keep it straightforward. If you have no endurance, that's because you don't do it this much. You're doing this. Describe the two events of putting one, maybe a bag over someone's head or your head, or two, drowning. What's the result? Death, basically, right? We kind of view that it's like your lungs are sad because there's no new air coming in or something like that. That's the way that we think about it, but it's really not. It's this process. The reality of it is that if you have no oxygen coming in for whatever reason, you go to pyruvate, and instead of going through this point, you stop, and you go back here. When people do that, or when that happens to people, better put, because I don't think anyone would purposely do that, I hope not at least. When that happens, the reason that happens is because they go brain dead, right? Isn't that the story? If you're underwater for so long, the longer you're underwater, the more opportunity for being brain dead. The reality of it is that your brain needs a ton of energy to fire its neurons. It's just the reality of it. Your muscles need a ton of energy to fire their neurons. If you don't have that energy present, it's the tissue there starts to deteriorate. The tissue dies. Your neurons die. So ATP is a requirement. So I have to have ATP. And the bulk of ATP is produced in this process. If you're not going into this process, into this mitochondria, then you're producing only 2 ATP. Now, to let the cat out the bag, as they say, this process down the line that we'll start talking about a little bit with tomorrow and Wednesday, this process through here actually results in a ton of ATP production. As far as the amount of ATP that's produced, the amount of ATP that's produced on this level is somewhere around 36 ATP. Now, that means your process of burning down the glucose could produce either 2 ATP only or could produce 38 ATP. Now, are you breathing now? Were you breathing yesterday? And the day before that and the day before that? And so was all of humanity since the dawn of the kingdom Animalia has this happened. In fact, we could go even further back, and it was like 2.5 billion years ago that this idea started to evolve. Its beginnings existed. But if we stick to just your lifetime and the fact that you breathed yesterday, or that you were breathing yesterday, that means that your body has a need for not only these 2 ATP, but for these other 36 over here. So your amount that you require is a ton. So if you're not able to be aerobic, you can't be doing it right. Have a good day. We'll continue on with this tomorrow and move into the mitochondria, I think.